Hello, good morning, good afternoon, and good evening. Welcome to No Food Live, wherever you are. I'm your host, Matthew Lang, joined by my co-host, Courtney Riggle. uh, Welcome to the fourth episode of the No Food series, where we take a deep dive into the world of food informatics and the Internet of Food. No Food airs live on the third Thursday of every month at this time. If this is your first time, welcome. If it's not, welcome back. And, you'll never miss an episode if you just hit subscribe right down below.
Hi, everyone. Thank you for joining us. We're so pleased to have you today. As Matthew mentioned, this is our fourth episode, so we're starting to pile them up. And we've already covered a series of diverse topics, most recently on how the semantic web of food enables alignment and interoperability of food systems ontologies and data. Our topic today is actually part one of a two-part series on food composition. And we, of course, invite you to join us for part two next month.
Yeah. That last episode on the semantic web of food was so cool. It's pretty neat, having been part of it since its inception. It's really amazing to watch it grow and diversify and actually to see what sticks and, what's just sort of been a flash in the pan. But today is something that is absolutely not a flash in the pan. Today is something that is going to continue forward because we're talking about molecules. We're talking about the actual essence of what's in our food that we eat every day. And if you ever wanted to know anything about discovering molecules inside of food, you'd be lucky to talk to any one of our three guests today. But we've collected three amazing food omics researchers right here under the No Food virtual roof.
Yeah, it's a very exciting opportunity. Our three esteemed guests today are all scientists at UC Davis. Selina Wang, give a little wave, Selina, from UC Davis Olive Center, is an expert on phenolics and post-harvest impacts on food. Daniela Barile, from the Food Science and Technology Department, is an expert on carbohydrate composition of foods. And Ameer Taha, also from the Food Science and Technology Department, is an expert on lipid interactions with the brain. Selina, Daniela, Ameer, we're excited to have you join us on No Food Live today and look forward to your presentations. And more so, we look forward to discussing how our collective work streams and outputs might interoperate more effectively.
Right, so let's get started. First up, we have Dr. Selina Wang, from a cooperative extension specialist for fruit and vegetable processing in the Department of Food Science and Technology at UC Davis. She's also the research director for the Olive Center at UC Davis. Dr. Wang works on issues to material purity and quality, shelf life, and chemical changes during processing and storage. In recent years, Dr. Wang has also worked on chemistry and sensory correlation, byproducts management and upcycling, and analytical methods for both olives and olive oil. Welcome, Dr. Wang. Selina. Before we jump into your presentation, though, I wonder if you might tell us a little bit, for our audience members who don't know exactly how the university system works, what is a cooperative extension specialist, and what's your role at the university?
Thank you. Happy to share that. My role as a C specialist is very similar to those our faculty members in the department, such as professors. We conduct research, service, and our type of teaching is through extension. So teaching with regulators and industry professionals. And
Great.
and we mentor graduate students and students the same way.
The same way as professors.
Yes.
That's great. And so you're really interacting with industry and a lot of their issues then, and at the molecular level, if you will.
Yeah, very mission-driven type of research.
Terrific. Thanks. Well, great. Well, I know you brought a few slides, so I'll get out of your way, and looking forward to hearing more.
Great. Thank you. Really happy to be here with my colleagues today to share some of the research that my lab has been doing. And I will start with this slide, which I think everyone is familiar with. Basically, is one form or another where we have the input in the farm and all the way till in getting the hand of consumers. So farm to fork is a similar analogy that you have heard. So starting on the left side, you see the input and production and processing and transport, storage, handling, and more value-added processing, and more transport and marketing and distribution, and that gets to the end user. For the sake of this today's talk, I'll focusing more on the left side. So what happens with harvest, what happened during processing, and what happens with storage. And I'll give you an example of each. And I think what I would like to highlight is that how the potential health benefits, flavors, and taste of food are affected by each of these steps. At the same time, we're also interested in production efficiency in such a way that there will be less byproducts that need to be recycled or upcycled. So this is a structure of just the basic phenol moiety. For many of the foods we study in the lab, we measure the phenolics, which is related to this molecule. And they're important for the health benefits of food, but also for the taste. It often correlates with some bitterness and astringency in the foods. We also like to look at volatiles, which are related to the smell of the food, which is important. I believe these are important chemical parameters to measure because consumers prefer foods that are good for them, but they also want them to taste good and smell good. So just give you some examples of the phenolics that you may encounter on your day-to-day life. So this is a structure of catechin, which you may or may not be familiar with, but I'm certain you have consumed it at some point. It's found in cocoa, chocolate, and green tea, or some different types of tea. And also in grape seeds has a high concentration on this. And we have some data to support this in my lab. And there's another example of phenol. And then you can see they all have the benzene ring that's attached to the OH. That's what makes these molecules a part of the phenolics class. So caffeic acid, obviously, you can find that in coffee. It's also found in a lot of herbs such as sage and thyme. It also has a good concentration found in argan oil, which people use sometimes on their skin and on their hair. And here's the largest molecule of this whole picture, which is oleuropein. And that is found in, of course, you can guess, from olives. It's especially high in concentration in green olives. So olives, as they mature, they go from green to purple and to really black or dark at the end of their maturation. So oleuropein is mostly in the green olives when they're still not super ripe. And this is the reason why you cannot, or maybe you should not, if you see an olive tree and just pick an olive and pop it into your mouth because it's extremely bitter. But these are important compounds that's in our food. And I'll talk a little bit about oleuropein, but more in general, the overall phenolics. So starting with olives. So this is an example of the work that we did in my lab in the last 4 or 5 years, basically. We look at these 3 most common cultivars planted in California. There are Albertina, Albersana, and Koroniki. And then the reason why they're most popular in California is because they're good for super high density plantation that can be mechanically pruned and mechanical harvested. And on the x-axis of this picture, you can see that our measurement of time here, what we use is days after flowering. And on the y-axis is oil accumulation. So we look at how fast the oil are accumulated in 3 different cultivars along the days after days of flowering. And this is important because the farmers who grow olives, they're paid by the oil content that's in their fruit. So this is very important regarding how much they can make from this year's crop. And also from the farmer's perspective, their timing of harvest is important. And then it's different for different cultivars. You can see that you will want to harvest Arbequina earlier, then you will harvest Harvestana, and then you will want to harvest Koroniki the latest, as the oil accumulates a little slower compared to other cultivars. But oil content, or EO, is not the only thing that we want to care about. We also care about the phenolics that's related to health, and also the flavor of the oil. And then as you can see, the trend is so clear, and it's not so consistent amongst the 3 different cultivars. And they usually go up and they come back down. So if you want to maximize phenolics content in the oil, you want to harvest at the time where it peaks. So, and you can see it's different for Arbequina, Arbestana, and Koroniki. So it is important for the farmers to take these factors into consideration and then make a decision that is reasonable for their own orchard and their own company. And that's not the decision that's made by me, or I don't have input on the decisions that they make. But I do believe that it is our job to help them to make educated decision by providing this kind of data. So we were really happy to do this kind of work for the first time, basically in California. And now we're building up this information because you can see that there's seasonal differences as well. This result, what you're seeing here, is the average from 3 different harvesting seasons. So this is how harvesting is important to impact the chemical composition of the oil that we consumed. And now moving on from harvesting, we're going to talk a little bit about how processing can also have similar effect. Again, I'll take an example of olives and olive oil. So in this case, we looked at the impact of the different grit size. So for the crusher, so we have 2 different grit sizes, a smaller one and a large one. So that's the only thing you have to know here. And then you can see there's 0 ppm and 500 ppm. That's with the addition of enzyme. So one, we added pectinase as enzyme during processing. One has no enzyme. And then you can see with smaller grit size, that's G1, and enzyme, you are able to recover more oil in green fruit. So remember, green fruit is less ripe. And we saw similar but less significant impact with more ripe fruit. So since enzymes are not exactly cheap, a processor probably want to invest that in more green fruit and use smaller grit size to get the most oil. Now we remember from the earlier slide that we also want to care about the phenolics. And the good news here is that you can actually maximize that at the same time with enzyme and smaller grit size. Now with that, we did another similar trial, but now this time we included even more variables. So this is on the left side, you can see that's an example of the grit size that I was talking about. On the left side, you have this really small grit size, and on the right side, you have the large grit size. And this time we included the impact of processing speed. So the crusher rotates, and then we want to see what impact does it make when we make the process, the grit goes faster. And then the reason why we have considered these kind of different processing parameters, such as the grit size, the enzyme, and then the speed, is that we're trying to do this kind of research in such a way that the processors don't have to invest millions and millions of dollars to change the equipment that they already have. Right, so changing the grit size and the speed is something that they can easily do instead of buying additional equipment and then adding to their investment. So what we see here is consistent with what we saw earlier, that the grit size, the smaller grit size, which is the 4.5 millimeter, gave the best extraction efficiency and the most phenolics. And faster rotational speed also give the best extraction and total phenols. This is good news, and it makes sense because smaller grit size and faster speed basically increase the cell wall rupture, so the more oils are able to be released from the oil vacuum in the cell. So we're happy with this result because it gives a processor information to make their own decision during processing. And they don't have to sacrifice phenols with yield or vice versa. So now we talked about harvesting and we talk about processing. So the last point I want to make is storage. So it's post-harvest. In this case, I'll take an example of walnuts. So this is a complicated, well, actually not so complicated plots, but there's a lot of data behind this. So in this case, we look at two different most common cultivars of walnuts. They're Howard and Chandler. And we put them during storage and we analyze samples during different time points. And we basically see how all the chemical parameters change with time. And we found that there's significant correlation between phenolics and different volatiles. And it's negative correlation, of course. So when the phenols decrease, there's undesirable volatiles that's related to oxidation that increases. And you can see basically they're on the opposite end. And there's also other compounds that's involved, such as fatty acids and terpenes and tocopherols, peroxides. But the correlation between the phenols and the volatiles were the most clear and significant. And just zero in on this a little bit. So here is some of the volatiles that we identified and quantified. And here's the heat map of the results. And you see that we analyzed Chandler and we analyzed Howard for 28 weeks. And the blue represents a lower concentration where red represents higher concentration. And you can see Howard oxidized faster than Chandler because you can see at 28, there's a lot of these oxidized volatiles, which suggests that if lowering the storage temperature costs money for the processors, they would want to make that investment for their Howard cultivar before they invest that for their Chandler cultivar because Howard oxidizes faster. And we're also interested in developing rapid chemical tools to not only detect oxidation, but potentially predict shelf life. So this is a step that helps us to get there, understanding how the chemical composition change with oxidation during storage. And not just oxidation, there's other chemical reactions that take place as well. And we're using this dendrogram, we were able to actually separate these samples that we analyze really well to the dark blue. Basically, there are samples that are not oxidized at all, to the lighter blue where they're mildly oxidized, to the orange, they are very oxidized. And they're all found Howard at the end of the sampling time. And then we're able to analyze this data a little further and basically zero in on the most important and significant compounds that we need to focus. So now we're in the process of narrow down this even further by collecting more samples. And then that can really help us to develop chemical tools to zero in on the really important chemical markers that are associated with these undesirable changes during processing. So I hope that was a good quick overview on how harvesting, processing, and storage can affect the chemical compositions of the food that we consume. Thank you.
Wow, thanks, Selina. That was super interesting, and obviously there are so many different leverage points throughout the supply chain where both the nutritional content and the quality of the product can be impacted by, decision making. So here's a question for you: how do you envision, or can you envision, this kind of information about a whole variety of products being shared efficiently to the people who need them for decision making? Can you see that future?
Yes, of course. And the part of I think the bottleneck is that it's different for every food, right? So it's very difficult to do something like this and then we'll say, okay, you'll work for all the foods. And I've been able to leverage our experience with olives and olive oil because it's a high lipid food, right? And then apply that to other commodities such as avocados and walnuts and almonds and pistachio. But partially is because if they have similar chemical compositions, then we know the the learning curve is less steep. But if talking about something that have completely different matrix, then that part of the data collection and understanding at a deeper level would take some time. But because we have to do it really carefully, thoughtfully, and with thorough investigation, right?
Yeah, absolutely. Thank you, Selina. So now let's shift our focus to our next guest, Professor Daniela Barile. Originally from Italy, Daniela is a professor and food chemist in the Department of Food Science and Technology at the University of California, Davis. Daniela's lab researches the chemical and biological properties of food components to generate novel ingredients that improve human health. And Daniela, can you tell us about your current initiatives and the collaborative network that you've developed with members of the food industry?
Yeah, do you want me to address that question before sharing the slides or after?
However you think it fits together best, but we're interested in your slides as well. So
yeah, so we do work a lot with industry because it's the best way to really understand what the industry needs, right? What are the problems? What are the molecules that they don't have the tools to measure and yet are the most relevant molecules to improve human health? So my research is really focused on human health, as you will see in the slides. And so we do have a network of food companies spanning many different domains. It's milk, but it's also fruits and vegetables, and even newer technology using recombinant organism to produce bioactive molecules. And that keeps us on our toes to always be pushing the envelope to the next level of what's the next molecule to improve human health. Great. So talking about human health, well, there's a crisis besides COVID-19. There is another global health crisis that has been happening, and as we see in the pictures from the cover of the Time, has been happening earlier and earlier in life. So now metabolic disorders like obesity and diabetes have an early onset in children and teenagers and young adults. And scientists have been puzzled about this rather recent insurgence of this type of diseases. The simplistic way was to look at these in terms of the food intake and the reduce energy expenditure, sedentary life, and just like diet-induced diseases. But actually the picture is a little more complicated, and with advances in studying the gut microbiota, which is the collection of trillions of bacteria that we harbor in our intestine, well, we and like many scientists internationally really discover a link between this type of metabolic diseases and alterations in the microbiota early in life. Think C-section, think early antibiotics intervention, think incorrect diets. And so as you can see in the cartoon in the right side of the slide, the gut microbiota, it's actually linked to the brain, communicates with the brain through the gut-brain axis. And if there's a early disruptions in the type of bacteria that you have in the gut, there can be a disruption because those are involved in regulating the body weight. So just remember this because we'll talk about it again. So we talked about the bacteria. So the gut microbiota plays many important roles in the human body. So feeding for health can take some unexpected directory, meaning we should pay more attention to what we eat to feed the bacteria in our gut compared to what we have been doing, feeding ourselves and counting calories when trying to lose weight. Because as we see in these slides, the gut microbiota, the effects are not just restricted to the intestine, but in fact, they affect the whole body, right? From gut-brain axis, behavior, feeding habits, behavior, immune system, liver, insulin, defense against pathogens. So many important activity which determine our health or disease status. So our research goal is to identify the food molecules that can modulate the composition of the gut microbiota. So make sure that we have desirable protective bacteria instead of pro-inflammatory bacteria. It's not an easy task, and we can ask ourselves, where do we start from looking at food molecules? Well, the answer we found is to start from the very beginning of life and start looking at the first food. There is already strong evidence that breastfed infants have some supercharged health benefits compared to infants that were fed infant formula in terms of breast milk offering protection against many infections, and even breastfed babies having lower rates of obesity and metabolic disorders, which is exactly what we're looking for to understand, right? So that's why breast milk was a really ideal food to start from. The bioactive molecules of breast milk have been retained and actually amplified over more than 100 million years, selected by evolution. So they must be really doing something good for human health. As we can see in the slides on the left, there's a snapshot of milk composition. We have macronutrients and micronutrients. So macronutrients are well known. There's databases about that, and they affect the growth and the body composition. And then we have micronutrient status, elements, vitamins. But what's really, really important, and what is most unknown, embarrassingly, about breast milk, it's the category bioactive components. And these bioactive molecules are in charge of determining gut health, neurodevelopment, and immune system development. So you can see how we need most knowledge on the molecules that are doing the biggest job for the developing body, and those are the ones we really don't know about. But actually, thanks to some coordinated effort led by Dr. Bruce German, the picture up left, who was my mentor here at UC Davis when I came as a student, he founded the International Milk Genomic Consortium. And so at UC Davis, a lot of progress has been done in decoding and deciphering the structure of these molecules that can make all these fantastic activities: gut health, neurodevelopment, immune development, protection against pathogens. These molecules are complex indigestible carbohydrate. You see a cartoon in the slide. They're called human milk oligosaccharides. It's interesting because they are not used as nutrients by the infant, non-nutritional, 0 calorie. But instead, they feed the bacteria in the gut, and so they help establish a healthy commensal bacteria. So very important molecules, but we don't know very much about them yet. They are complicated structures, and how does it work? In very like 30 seconds, years and years of science done by microbiologists. Well, nature came up with a very ingenious strategy. All the linkages, the things written in red that unites the building block of these molecules, they're so complicated that only few bacteria, especially strains of bifidobacteria, which are beneficial, which co-evolve with the oligosaccharides in human milk, are able to cleave, and so they possess the scissors to cleave the monosaccharides and so use them for their own energy, and then can multiply and colonize the gut. So pathogens will actually starve because they're not able to access this carbon source, which is the only carbon source in milk. So it's a really precise and design strategy. So clearly there's a lot of potential, and we did clinical studies showing that human milk oligosaccharides can really save lives in case of premature infants. Lots of knowledge here, but what about the industrialization? How do we bring it to the larger population that needs these molecules, except besides breastfed infants? So we need to translate this knowledge and bring it to practice. As we mentioned at the beginning, there's really a critical need for food-derived solution to eliminate pathogens, because the pathogens are now becoming resistant to antibiotics. And so antibiotics and the pharma industry cannot help there. We need food molecules. So what we're looking is, we have these check marks like, we know what human milk can do. We know the molecules now, and we want to find them in other food that are more available, and especially food streams. So if we can find the oligosaccharides that can eliminate pathogens, improve the growth of desirable bacteria, reduce inflammatory diseases, while keeping in mind that we want to develop sustainable processes and reduce agricultural waste, well, that's our trajectory. So how do we get there? We need to build a detailed understanding of food and agricultural streams. We need to catalog many, many foods for the bioactive oligosaccharides, which are small molecules. And then we need to bring this knowledge to practice, as Courtney was mentioned, by collaborating with the food industry. How do we do that? Chemistry is the key to help solve this problem. And so with chemistry, we can transform food waste into gut microbiota modulators molecules. So we do that by using tools like mass spectrometry. You can see one example in the top right of the slides. And then we create bioinformatics oligosaccharides libraries, which is like digitizing food bioactives, because we are creating the fingerprint of each food. And it doesn't look like a fingerprint. It looks like that series of color peaks that you see in the slides, which say this particular food contains all of these oligosaccharides in this concentration. It wasn't easy. It took us many years to actually do this process for various types of mammalian milks, which was the first obvious food source to look into, quite similar to human milk. So we found oligosaccharides. You can see like a database looks like a table with a lot of information, not very easy to use for the non-expert. So we collaborate with Agilent Technology to incorporate all our information into a software. And now new scientists approaching the field, if they use the same tools, they can actually see popping up the structures like circle in yellow in the screenshot in real time. They inject a sample and they get the structure popping up on the screen. So this is how knowledge can accelerate discoveries. Just to conclude and give you one example, we talked at the beginning about obesity, metabolic disorders, and the gut-brain axis. This is one application in collaboration with BASF, which is a large chemical company. We gain access to one of the key prototypic oligosaccharides molecules that are in human milk, and we were able to use it in an animal study where we induced obesity, induced metabolic disorder, interrupted the communication between the gut and the brain, and then these oligosaccharides was able to restore the system, restore the communication, change the gut microbiota, and reduce all the disturbances of metabolic diseases. So very practical application. I talked about bovine. We study actually many types of mammals. We are understanding through funding by the USDA the influence of breed, the influence of diet to increase the levels of these molecules for extraction and transformation in ingredients. And then we looked at also plants and pleasurable fluids like coffee and wine. And we found oligosaccharides there. The next step then, which we're doing, is optimize bio-guided processing to preserve the oligosaccharides and extract them from the waste stream, like coffee beans, coffee grounds, grape pomace, winery waste, and many other types of plants. And with this, I'll conclude, and I'll look forward to the discussion.
Wow. Ottimo. Grazie mille, professoressa. Let me ask you this. You just gave us so much information. So you highlighted the waste streams, or what we might desire to call potential, what would otherwise be waste streams, because nobody wants to consume waste, and the entire sort of processing stream. And building on what Selina was saying, it seems like we could find some generalities in terms of understanding processing lengths and what happens along those processes and the types of molecules that are extracted. but there's a lot of work to be done. If we could imagine a database of all of these molecules coming off and appreciating the breeds, the varieties, the processing methods, and how those alter the molecules, what in your mind would be the bottleneck to keeping that from happening? Is it naming conventions for molecules? Is it the actual building of the database? Is it unwillingness to share information? What do you think is like the bottleneck that's currently sort of keeping things from happening, and why we don't have this technology already?
Yeah, great question. Not an easy answer. A little bit of everything you listed, in the sense, some data, especially working with companies, will remain proprietaries, even though we do push for publication and for releasing information for the public. The other point you made, nomenclature, like the naming convention, it is true, because when different groups on different area, different part of the world use different instruments, everybody will publish in English, but the actual naming of the structures will be very, very different. And so even though we specialize, the scientists can understand all the different convention, but you can't translate this into database automatically. So there is a lot of manual curation that needs to happen. And right now, this effort, it's not funded, not yet even for human milk, right? We need to first do that for human milk, and then do it for bovine milk, and then do it for coffee and almond and chickpeas and all of the other plants that have the potential. They have similarities in the building blocks of the oligosaccharides to the gold standard.
That's great. And just to give a quick shout out, you mentioned Dr. Bruce German as sort of spearheading some of this investigation into human milk. He'll be joining us next month for Food Composition Part 2. We're very much looking forward to having him, and that's pretty exciting. So thank you, Daniela. We'll come back to you for a deeper dive into some of this after we hear from Dr. Taha. So Dr. Taha, Ameer Taha, is an associate professor and a food chemist in the Department of Food Science and Technology at UC Davis. And whereas Dr. Wang spoke about phenolics and Dr. Barile spoke about oligosaccharides, Dr. Taha is here to tell us about lipids. And those are the molecules that people generally refer to as fats and oils. So Professor Taha, Ameer, can you walk us through lipids in the food supply? And I think you're also going to tell us a little bit about their impact on brain. Please take it away.
Sure. Thanks, Matthew. So lipids are, a lot of you are familiar with lipids, also commonly known as fats. They're in the food supply, but importantly, they're also in the brain.
Sorry, Ameer, I just want to interrupt you 1 second. We're looking at the design view of your slides. Can you pop it back into presenter mode?
Oh, yeah. Let me do that now. Do you see my slides? Okay.
We still see the design view.
Oh, I see. Oh, got it. Okay. Presenter mode. Yeah. How about now?
Yeah, that's perfect. Thank you. Yep. I'm out of here. Bye.
Okay. All right. So lipids, they're in food. They're delicious, as most of you know, but they're also in our heads. They're in the brain. So today I'm going to talk about some of the impact of how oxidized lipids can affect the brain. Now, plant oils used in food processing are a source of fat, and they are mostly high in a particular fatty acid or a fat called linoleic acid, which I'll abbreviate here as LA, linoleic acid. Now, you can appreciate from the picture here that a lot of the oils you see are pretty high in LA. So sunflower oil is about 75% by percent weight. Corn oil is 56%, and olive oil has the least. But you can see that this fatty acid is quite abundant in oils used in food processing, which is why it's fairly abundant in the food supply. So this slide here shows on the y-axis is the percent of the U.S. population on a certain energy level of intake of linoleic acid, or LA, and the x-axis shows that energy level. So let me put this into perspective. 0.5% energy is the minimum requirement needed for just existing. This is the minimum amount needed. Now, this happens to be a little bit less than pre-agricultural diets. So before the 1900s, linoleic acid in the food supply was pretty low. So it was around 2% energy. So pre-industrial times, pre-agricultural diets, and I'll talk a little bit about what that means. It used to be around 2% energy, which was fine because the minimum needed is 0.5%. So 2% was, although it's 4 times the minimum requirement, it's still fairly low. Nowadays, with the advent of soybean agriculture and with the advent of soybean agriculture resulting in soybean oil, as well as other types of seed oils containing high linoleic acid, current average intake of LA is over 5%. Actually, the US average now is around 7%, but as you can see here, some 20% of the population is above 10% energy. 5% is 10 times the minimum requirement. 7% is much higher, 14 times higher. So you can appreciate that there's a lot of linoleic acid in the food supply. Now, LA, just like any fats, can oxidize. What this means is that you can add an oxygen to the molecule. This is what oxidation is. And in doing so, you turn it into, in the brain, you can turn it into a signaling molecule. Now, lipid oxidation, or LA oxidation in this case, is really important for food industry because when lipids oxidize, this is how we can enjoy French fries, because in French fries, the French fry taste and smell is in part due to lipid oxidation. This is how we can enjoy food. On the other hand, this is how food goes rancid, because too much oxidation can cause rancidity, can lead to rancidity. In humans or in the brain, we actually have enzymes that can oxidize linoleic acid or other fatty acids to make what's called oxidized linoleic acid metabolites. So the difference between food, between cooking and the brain is simply that in cooking, you heat the food to oxidize fats. The brain has enzymes, certain proteins that can actually do it for you. So you don't need to really cook your brain to oxidize them. You have enzymes that can do it at 37 degrees Celsius. Thank goodness. When you oxidize fats or LA, you get oxidized linoleic acid metabolites. These are so diverse in structure. There's yearly there's new molecules being discovered. So you can add an oxygen here, you can add it there. It gives you a different molecule. You can add it in a different form. So here it's called an epoxide. You get a different molecule. So there's hundreds of these molecules that are created in the brain. And our question was, is this good or bad? That's a primary question. And the answer, it's complicated. It depends. It depends on the stage of life. And I'm just going to show you some few slides to highlight what I mean. It all started when we, back in 2019, we sort of observed that there's something different between the young brain and the adult brain. This is in rats. In young rats, in young rats, so what you see here is a pie chart. The two left pie charts are in young rats. So we call them pups here, males and females. And then on the right side, this is in male adult rats. We have not done females yet, but we're doing it. But there's one notable difference. If you look at this gray area here, this dark gray part of the chart of the pie, in both male and female pups, young, this is 0 to 1 day old rats, baby rats, basically. Half of the oxidized lipids in the brain, which we call oxylipids, another way to say it, are made of these oxalams. So specifically the oxidized lipids coming from LA or linoleic acid. If you compare this to the adult brain has about 7%. oxalams. So this is a huge difference. A developed brain has roughly 7 times less oxalams compared to a developing brain, a baby brain. Now, this led us to think that this may be important for development. So what we did was we took some neurons from baby rats and we exposed them to one of the oxalams, one of the most abundant oxalams in the brain, called 13-hydroxyoctadecadienoic acid. Say that 10 times. So when we applied this to neurons, we found baby neurons, basically, so neurons coming from baby rats. We found that it actually increased axonal outgrowth. So the neurons were sort of trying to connect with others, and the axons, so this part of the neuron was extended. Now, we don't know yet whether this is a good or bad thing. We're actually looking into that. It's one thing to increase axonal outgrowth. That's generally a good thing. But the next question is, do they connect with other axons? Because connectivity is really critical. And that's one of the things we're trying to explore. So you can see this as partly good news for now until further evidence comes out. And we're currently studying the implication of these findings on brain development. We're actively doing studies at the moment. I don't have new data on this. We just finished a big round of our large study trying to examine what these do, but hopefully in about a year we'll figure it out more. Our key question is, more of this in the brain, is it good or bad? And I don't have the answer yet. But at least when you apply it at physiological levels, it seems to increase neuronal outgrowth. In adults, so then the question is, what about adult rats? Are too much oxalams in the brain good or bad? So one way to change oxalams in the brain, because I told you earlier they come from LA, linoleic acid. So there's many enzymes in the brain that make them. So LA gets in and it gets converted into these oxalams, and we've shown that to occur. In a previous study, we lowered linoleic acid and we found that actually the brain oxalams were reduced. So here our question was, if I lower dietary LA intake, so LA in the food, effectively banning French fries to people, that's one way to do it. Well, not highly effective, but it does lower it a little bit. If I lower LA in the food, would I increase the resilience of the brain to inflammation? So this is a complicated slide, but what we did was we fed rats a low or high LA diet for 15 weeks. So low is about 0.5% LA. So this is close to the minimum energy requirements needed for rats. And high, this was about 5%. So going back to that slide, in the US, most of us are above 5% energy LA, in fact, around 7%. And after 15 weeks, we challenged we directly injected into the brain artificial cerebrospinal fluid to rats on either diet, or lipopolysaccharide. So this is bacterial lipopolysaccharide. It's a toxin, and we injected it directly into the brain. So this is the stuff that when you're sick, it's produced, and it's one of the reasons why people feel sick after a cold or a similar infection. So what we did was we directly infused this into the brain, and we found that, so on the y-axis are markers of inflammation, prostaglandin E2 and cyclooxygenase activity. Incidentally, these are the same targets for ibuprofen. So when you get a headache, you would take ibuprofen, and the idea is that it reduces cyclooxygenase activity, and in doing so, you reduce prostaglandin E2 production. What we found was LPS increased prostaglandins and cyclooxygenase activity in rats on the 5% energy LA diet, so similar to average intakes, broadly speaking. But when we lowered the LA, this effect was blunted. So with LPS, you don't see this increase in prostaglandins or cyclooxygenase here. So this led us to conclude that there's some protection to be gained by lowering LA in adult rats. And I haven't shown the data here, but this corresponded to a significant reduction in brain oxalams as well when we lower LA. So what about humans, specifically elderly humans? This is in rats. Rats and humans are a bit different, very different. In humans, Circulating oxalams, we reported recently that they're associated with poor executive function, but primarily in people with vascular cognitive impairment. So let me take you, if you look at the right side of this slide, on the y-axis you have executive score, which is a measure of executive function. On the x-axis you have what we call the diol to epoxide ratio. So these are ratios of LA metabolites or oxalams in the blood. And basically what this shows is that if you don't have cognitive impairment, if you're an elderly with no cognitive impairment, so these are different oxalams here shown on the top and bottom. If you're an elderly with no cognitive impairment, these don't really correlate much to your executive score. On the other hand, if you have vascular cognitive impairment caused by vascular disease, which is a precursor to, which is a risk factor for Alzheimer's disease, actually, we found that increased concentrations of these oxalams is associated with poor executive function. So this was work done in collaboration with Dr. Walter Swartweiger from Sunnybrook Research Institute in Toronto. This basically tells us that too much oxalams, especially in at-risk individuals with vascular disease, may not be really good for cognition. And we're following up on this in other studies. So what's the take-home point? A couple of things. One, oxalams, which are oxidized linoleic acid metabolites, they seem to be quite important for neurodevelopment. I mean, just think of the fact that half of the brain oxidized lipids in baby rats are made of these oxalams. And we're showing that they regulate neuronal outgrowth. So they must be important, but that is yet to be confirmed. In adult rats, they're associated with brain inflammation. And in humans with cognitive impairment, in humans, they're associated with executive dysfunction, particularly in people with vascular disease. So therefore, the effects are not so straightforward. They tend to vary with age and disease vulnerability. And we're still trying to explore mechanistically how and why and further what these molecules are doing. That's all I've got.
Wow. You're freaking me out, man. I'm scared of our food supply right now. Like, so I just want to make sure I understand what you just said. So a big chunk of our diet are these lipids that contain linoleic acid, and when those get oxidized, they could essentially impair our executive function as we age and develop age-associated diseases like vascular disease. Is that what you're saying?
The data seems to point to that, yes.
And do these oxidized lipids also play a role in causing that vascular disease?
So that's a great question. We don't know whether it's a cause or due to vascular disease itself. So there could be underlying genetic factors that predispose you to vascular disease, and we're seeing this as a signal, which is why we think it's a nice biomarker at the very least. But this is something we're trying to look into further, actually.
So, but I mean, I don't know. It seems maybe the laboratory evidence and the clinical evidence hasn't played out yet, but it seems we know a lot of these oils are in the diets of people who are in obesogenic diets, essentially, right? And that people who have obesity have a higher propensity for these types of vascular disease. And now what you're saying is not only do they have essentially vascular disease, but potentially down the road cognitive impairment as a result of these circulating oxidized lipids.
Yeah, I think it's a vicious cycle that ends up being created, feeding into one another. The vascular disease itself can impair cognition, and then you have these oxidized lipids being generated, and I think it just creates a very uncomfortable chemical scenario leading to dementia down the line.
Right.
And individuals with vascular disease do tend to, there's a higher risk for them developing Alzheimer's as they age.
Right. Wow. So why don't we get the rest of the group in here, and maybe I just want to sort of go around the table since we're already sort of on the subject, Ameer. I think it would be good if we could just sort of daydream a little bit for a second here, right? And talk about, well, if you were in charge of the food supply and information about food, and you could wave a wand and say, gosh, based on what I know, here's what we would be doing with foods and sort of with you in Ameer around lipids, and what would you say would be something interesting? Would you move people's diets? Would you do more analysis? Like if you had the $10 billion to spend on this, what would you do?
I would, leaning on our findings in rats, where the hypothesized mechanism is that these linoleic acid oxidized metabolites are driving inflammation in the brain. And when we lowered it in the animals, we found that inflammation was reduced, comparable to given someone, simplistically speaking, given someone an ibuprofen. But so if I had the magic wand, I would probably test the effects of lowering linoleic acid through food, and observe whether this actually leads to benefits in cognition or related disorders, basically.
How about you, Selina? What would you do if you had the magic wand?
Well, so earlier we talked about my role as a C specialist, and part of that is to educate and assist farmers and processors and consumers. So I would like to use the information that we have gathered and make that easily digestible and accessible for those who are farming, who are processing, and who are dealing with the supply chain. And then the retailers, I think the education for retailers is also important because they sort of serve as a gatekeeper for many of the foods that we consume. And a part of the work that I didn't talk about is food authenticity, right? So the transparency of that part of supply chain is also important. And I believe that we have the technology, and a part of the work that needs to be done is the integration and adoption of that, and to make the information much more accessible for those that need it the most.
Mm-hmm. Daniela?
$10 million?
Billion with a B.
Well, I think there's a need to have a center around really having people with a strong computational background and chemists working together, because right now we are assembling lots of data in Excel and manually curating the literature, but then there's always a block to get it into a form that's queryable, where you create really fancy visual that other people can use, and they can look at this data set and see, oh, I have this other data. How can I contribute to your data set? And we're chemists. We don't have those skills. So there's really a need to hire an army of students, or like trained students in both chemistry and computational technology, AI, and all of these things that really gives you the capability to look at the food from the beginning to when it's harvested, how the composition is changing, when it's processed, when it goes into the consumer, and then do clinical study and have the ability to have the funding to really study all the biological fluids that are coming from people, because we have freezers full of poops and urine and bloods, and it's really hard to do everything. We are a single lab right now. And I also would like to work more with Ameer to understand, actually have him analyze our diets, the high-fat diets we feed to animals, because to me, everything is really linked. like the lipids and the inflammation and the obesity. You just added an interesting piece to the puzzle there. So we
So
need to
yeah,
study
we
food.
we
Yeah.
we could end up the animals that are in our food supply also have a diet, right? And I mean, we could have demented cows running around out there because they're consuming, food.
Not the mad cow, but
But did I just hear you say, Daniela? Did I just hear you say that there's a need for food informatics? Did I really hear you say that? 'Cause
for
isn't it part of? I
Well,
thought it was part
I
of the
want to
training of
ask.
food science. Sorry.
not yet.
Let
Not
me ask
yet.
a question on the same stream, though. So, for each of you, how do people currently access your information, and who are those people? Because it's clearly not getting to all the people that need to be getting to it. So, Daniela talked a little bit about how some of the research that she works on is proprietary, not all of it. But Ameer and Selina, how do people access your research, and who isn't accessing it that should be?
Selina, I'll go after
So,
you.
Ameer?
So through publications, I try to publish in open access journals when possible, but I also work closely with commodity boards and regularly give educational workshops, seminars, and scientific. I write two different types of reports, one for publication that's very scientific, and I basically translate that to layman terms and submit that to the commodity boards and create educational toolkits that is easy for them to access. And also, I want to be very direct. For example, if you're processing faster using grid size, smaller grid size, you're going to get better oil and more oil, right? So something very easy and for them to adapt. So those are the ways that I work on. In terms of what do I do with all the data that I'm sitting on? I would love to join this center that we're speaking of, and basically to put all that together. We love that.
So we follow similar methods of knowledge dissemination, and I actually try to put a lot of this stuff into my teaching material at UC Davis when I teach food chemistry or or other classes. But ultimately, a publication is the primary form, and then outreach and conferences. And we're in touch with industry also, trying to, if they have any questions, I try to address them in this regard in terms of LA composition of the oils or the food. And one thing which I hope we are working on, and I think this will inevitably bring us all together, is how processing affects LA itself in food. We've started to work on that. So the phenolics have a role, in terms of preserving it in a way, or not, or in some potentially under certain circumstances, oxidizing it. And this, we don't know a whole lot about that. And I think down the line, I think the important thing is to understand how oxidation occurs in the food itself, what products are produced, and how these enter the mammalian system and ultimately the brain. And from what we've done so far, I just want to highlight that it's a bit complicated. It's not as easy as more heat means higher oxidized lipids. It's really, really complicated.
So, given that it's complicated, I'm just going to sort of go back to you with the same question that we asked Selina earlier. Can you imagine a database? And come around to each of you, but starting with you, Ameer, can you imagine like a database similar to like the USDA food composition database or something, where people, like a central authority, can come and find this kind of information, or where researchers like you can deposit information and see information from other researchers? Does such a thing like that exist for you, or would that be helpful?
I mean, I feel that would be incredibly helpful. Simply because we can effectively say, okay, processing or the way you harvest this fruit leads to this level of oxidation or LA in the diet. But the next question for which we want to share with the world is, what is it doing? Does it get in, and what does it do to physiology? And I think it's going to require more than just our lab to address these questions.
Right. How about you, Daniela?
Um, yeah, I want to mention there is an effort in the good direction because we are collaborating with Naomi Fukagawa, and she has invited our lab to perform analysis on various plant products for oligosaccharides, and then our
I just
results.
want to interrupt you. You said you're collaborating with Naomi Fukagawa. Can you tell us
Yeah.
a little bit about who she is, what she does?
Oh, she's in Beltsville. She's a director of the nutrition center there, and she has been working for several years on the food composition database. And because she realized the importance of oligosaccharides completely missing from that database, she recently invited my lab to look at different type of milk, like soy milk and almond milk and other types, to see what oligosaccharides are actually contained in that. Now, I don't have the results published there yet, but it's a step in the right direction to start having these molecules, taken into account and have visibility and be included in food compositions because they are powerful molecules. So it's a great, I think we are starting in the right direction.
Yeah, I would have to agree. Um, it seems like we're, potentially running out of room on food labels as well for people to be able to put all of the various things that are actually in their foods. Anything to add there, Selina?
Yeah, I think it's a challenge, right? Because, for example, so I'm currently working on trying to develop standards for avocado oil, and essentially that requires you to have all the chemical compositions of the authentic samples that needs to cover different cultivars, origins, and then with the change of climate, we're also noticing their fatty acid profile, for example, are changing, because often if the fruit is grown in a hotter region, their oleic acid level is going to be affected, and then their LA level is going to be affected. So that's going to be a continuing work, right? Not just we have this database and then now it's done. It's going to be evolving as our climate is changing, and in more different cultivars, more different breeds are going to be planted. That's also going to be adding to the complexity of all things.
Right. So they become essentially modifiers of maybe some kind of standardized, what we would expect under, some kind of X conditions that something was grown or raised in. And it could essentially be modified by variety or by growing conditions or storage or transportation or what have you. That's interesting.
You did mention, I think, that they're starting to have rapid field tests to actually test the fruit on the ground. Did I hear that correctly? And are those available? I mean, are they widespread yet?
For certain things that you can measure with good accuracy, but there's still a lot of things that in-field testing is challenging to get accurate and repeatable and reliable results.
And so that's interesting. Do you think that there is a potential role here for even companies developing technologies for these rapid field kinds of tests?
Yeah, I think in field and also in line doing processing, right? So those are
Right.
are certainly opportunities
Mm-hmm.
for people who are interested.
And this is true for all the molecules that we have mentioned. Like for lipids, for example, both Ameer and I are using high resolution mass spectrometry, which is necessary for studying this class of molecules at the lab. But these are very big and very expensive equipments with high vacuum. You can't just unplug them and bring them into another location, or even in a pilot plant, because they're very delicate. You can't have a splash of water on them. So it's important to work with companies like Agilent, in the case, of like mass spectrometry, to let them know these challenges so that they can start thinking about miniaturizing and scaling down the equipment. And that cannot be done for initial discovery, but once you have a food, you have avocado, you have a process that's established, then you are in a position to say, I know my markers. I'm not going to monitor everything. I'm going to monitor these key markers. Then I can see a device could be actually constructed for quality control purposes, for processing, and on the field as well.
And do you think the industry is receptive to looking for this, specific information and looking for this kind of tool?
Yeah, I think they're very, very interested because right now they have to constantly rely on different external labs, and the cost is adding up, and also the variability in monitoring batches is causing some trust issues, basically, because if you don't get the numbers that you're expecting, then you want to give up, right? Having control of the information, having your own little device that you can use always the same in your company, it's something that is very much necessary, and the industry knows that.
I just want to shift gears just a little bit. Rui writes in from YouTube because we're live, and I'll put this up on the screen. I try to read the food label, but most of the time I do not know what these chemicals do. So not only do we have an issue where we're not finding the chemicals, and so this is a translation problem, right? Like how do we get the word out about not just what these chemicals are, but their role both in food, maybe they're a preservative or they're a flavor molecule, and in your body once you consume them. How can we solve this problem? Anyone? I'm going to pick on you, Selina, since you're the extension specialist and you work with the industry the most.
I think it's about so much that we need to do and so much we need to undo. I think there's a lot of fear about having chemicals in food, in consumers, and I won't get into details, the history of that and how this has come about. But I think that part is important, right? People need to understand chemicals are not all bad, and there's a lot of chemicals in a lot of things, and also processing, right? Processing, people associate processing food is bad because sometimes they hear that, but processing is an important part of food safety. So I think there's a lot of that need to be undone so that people then can start accepting the actual true knowledge about food. And I think what you're doing here is great, right? So this kind of work is really important to convey the important message in a very clear and concise way that people can understand. So I don't think I have an answer to this question. That's well,
it's a big question. It's a big question.
An idea could be, since the consumers that are most interested in knowing more about food labels are likely also familiar with technology, to leverage the QR codes, which now you need everybody has a smartphone, so you can get the information. If that was linked to a database that not only contains the list and the structure, but also tells you a little bit about the health benefits, maybe, with the link to publication, but we need people like Selina to actually simplify it, right, for the general population and say, okay, this molecule is good for brain health, this molecule is good for such and such, backed by publication.
Right. I remember someone telling me at a previous IC Foods conference that they don't want chemicals in their foods, but they didn't realize that their food is actually made entirely of chemicals, and so we have to even, I think, improve our language and say, well, which kinds of chemicals, and I think that's actually in fact. Flavor 360 software writes, isn't all foods made of chemicals by definition? So yes, that's exactly right. How are we doing on time? It looks like we're pretty close here.
Oh yeah, I think we've about used it up. So
yeah. So you want to shut us down here, Courtney?
I do, and I know we had an announcement to make, so I'm looking for it.
Oh, right. Well, go ahead.
Well, before we do that, I just want to, of course, thank our wonderful panel, Selina, Danny, and Ameer, for sharing their work with us today, and kind of helping tease open some of these really important questions on, how do we really access this information and how do we make it digestible for people? And I think we have a lot of future conversations and future collaborative opportunities to explore here too. So thank you all. A little virtual clap here.
Yeah. And before we sign off, just to remind folks, our next No Food Live will be Food Composition Part 2. Better food composition data means better diets and better health. And we'll be taking a deep dive into some of the standardized databases that are both online and coming online. And we're very much looking forward to that. Don't forget, if you like the show and you want to stay in touch, you can click subscribe right down below, and you'll automatically be in touch with us and stay in the know. Bye, everyone.
Thank you, everybody. Bye-bye.
Bye.
Bye. Thank you.